Good morning again. The next session is on emerging technologies in characterizing pancreatitis. I am moderating this session. My name is Dr. Maysam Abu El-Haija from Cincinnati Children's. Joining me today in co-moderation is Dr. Chris Forsmark from the University of Florida. And we have a great session and speaker list. Please remember that you can include your questions in the chat format function, not necessarily on the Zoom chat. It's better to do it in the session chat. And this way it will be saved for 3 months, I was told, and the speakers could have the chance to interact with you and collaborate possibly. So I would like to start by introducing our first speaker, Dr. Aida Haptizion. And Aida wears 2 hats. She's tenured and full professor from the Stanford School of Medicine, and she's also the chief medical officer of Pfizer. Aida is talking to us on cutting-edge tools for immune phenotyping of pancreatitis.
Thank you to the organizers for this opportunity, and thank you, Maysam, for the introduction. So I will be speaking about, I have been tasked to talk about technologies, cutting technologies that we are using in pancreatitis, specifically from FACS to CyTOF to single-cell RNA, TCR sequencing, and codex. And in terms of conflict of interest, currently I'm on leave and at Pfizer, but I will be speaking about the work that was done at Stanford. I want to acknowledge the 3 postdocs that have worked on these technologies and how they applied them. So I would like to really acknowledge these talented postdocs, Dr. Bomi Lee, Dr. Murli Manohar, and Dr. Elena Jones. Elena has currently left a different path in career. She's actually joined McKinsey & Company Consulting. Murli is currently looking for a faculty position, so he's up for grabs. And Bomi is working on her career projects and grants, so be nice to her and you'll see what she's done. And she's also going to be looking for a job actually in the near future. I also would like to take this opportunity to thank Dr. Sohail Hussain during my transition, who has taken over and has been kind to mentor these very talented postdocs. So before I speak about the technologies, I would like to think that we shouldn't just think technologies for the sake of thinking of technology. I always like to think why. It's always to start from the biology, the question, and then with that, then we can try to choose which tools, and the context matters. The other thing is knowing the challenges and limitations of these technologies is very important. There are technical and non-technical challenges, as you know. Of course, other things that one needs to take factor, of course, is the cost and many of other things. Do you want a depth analysis? But that's going to, of course, cost more. But in addition, there is also an analytic bottleneck. So this is when we use these tools, this is a great way to have team science because you're going to need expertise in the different field. So what are the field? And when I mentioned in terms of the why, the biology that we want to address is, it's always good for me, at least, I think about the gaps in the field. We know that there is gap in the field because we don't understand quite well the natural history of disease. We need longitudinal studies to address heterogeneity in the disease, and especially those that are at risk. Who are the pancreatitis patients that will end up having recurrent attacks? Who are the ones that will progress to chronic pancreatitis? And who are the ones who will just have 1 attack and will do fine? So this is a very important question that is to be asked. And also there is limitation because access to tissues or pancreatic tissue, particularly in acute pancreatitis, is almost impossible. So, so we'll talk about how we are trying to delve into these questions and what we are doing with these technologies. Another thing, of course, challenge in the field, as you know, is diagnosis. Making early diagnosis, particularly in chronic pancreatitis, is a challenge and something that one needs to address. And also finding essentially less invasive biomarkers would be helpful. Therapy, of course, is a challenge. As you know, as much as it is very common disease, we still don't have any FDA-approved therapy for this disease. And also there are, of course, challenges there in terms of what outcomes to measure, et cetera. So I'd like to keep those in mind. So the translational aspect is important, and this is why mouse studies are also important. As much as we do, of course, human studies, mouse studies also have an important role, and that's what I'm going to be talking here in terms of the kinetic studies that one can do with the animal studies. You can knock out, and especially certain genes and functions, one could look using mouse models. In the human, what we are doing is working with the consortium. We are fortunate to have been part of the CPDPC, the NIH-U1 consortium, to be able to get some samples. And there's also future studies that we are working with Dr. Pandol's program project DOD, looking at some of these risk factors that lead to pancreatitis, acute and chronic pancreatitis, those that are involved with alcohol. And we've had interest in the smoking as well. And the other thing, of course, is how to access human tissue. And we have been fortunate to work with Dr. Melina Bellin at University of Minnesota to access patients that are ongoing total pancreatic islet autotransplantation. And there is also a future recent DOD grant that was granted to look, in fact, at systemic and also local areas. So here are the technologies that I will be briefly touching upon. I'm not going to go into details, and I give you the posters. All three postdocs had a poster of distinction. Congratulations to them, and please do attend their posters for specific details, but I'll be talking of these immune phenotyping approaches, and I'll just outline, as I mentioned, in the interest of time. So this is from Elena and Bomi's work. This is together with the CPDBC NIH consortium, and you'll see on the table the types of patients. And this is, as you know, the PROCEED study in this consortium is a very important prospective. I think it's one of its kind that had collected samples from these patients, and we were fortunate to get some of these serum samples as well as blood samples to apply these Luminex studies, and also Olink, which is the latest one in terms of the proteomics. And there is numbers that I have listed there in terms of how many analytes one can do. I just would like to remark, these are good tools for a discovery, not really for functional, etc., but they're very helpful when we don't know, especially when we don't understand the disease and how we can use these biomarkers. So this is just a glimpse at the poster that will be presented, and Bomi is going to be presenting this poster on Friday, looking at some of these differences. And so going back to the clinical question, we need to make sure we compare the right control. So for example, we look at control versus acute pancreatitis and chronic pancreatitis, but maybe that is not the best in terms of the biology or the question or clinical question we want to address. The next one, an important one here that I put, is because we would want to know early chronic pancreatitis versus recurrent acute pancreatitis. As you know, recurrent acute pancreatitis, there's fairly significant number of patients who already have chronic pancreatitis that have recurrent acute pancreatitis. So biomarkers that would help us differentiate these two would be helpful. And similarly, from chronic abdominal pain versus chronic pancreatitis would also be important. FACS is single cell analysis, is our bread and butter in immunology. And as you know, it's widely available. This is a cost efficient, and this is something that we've used in many of our studies that we were able to understand, and in terms of mechanisms, how immune cells function, and also pathogenesis of experimental acute pancreatitis. As you see, over the years, we've used flow cytometry to analyze and also understand these cellular interplays, whether it's macrophages and stellate cells, or the role of immune cells play in these disease models. From flow cytometry, however, as I mentioned, it's available, but it doesn't really go in much depth because of the limitation. So in terms of how we have also used it in human pancreatitis, this is Bomi's work that was published in JCI last year, is that for the first time, actually, she was able to show that this is, again, from the TPIAT samples that we received from the University of Minnesota, that in fact, there is immune responses that seem to differentiate. We used to think chronic pancreatitis is just one chronic pancreatitis, but here you see, even at the T cell levels, that there is difference between idiopathic and hereditary chronic pancreatitis. This is just an abstract, graphical abstract from her paper, showing that there are, in fact, differences even in the TCR or T cell repertoire, although we didn't do single cell at the time. This was just cDNA based, because this was first assay, because you don't want to delve into a very detailed analysis. And this is what I meant, the context matters, because we wanted to first look, is there a difference, overall difference? And then we wanted to go more in depth, and I'll show you some of the data that she has been able to generate. So from FACS going to CyTOF, CyTOF is a mass cytometry, although it was not created at Stanford. In fact, it was first created at the University of Toronto, but it was pioneered by the Nolan Lab at Stanford, and there have been many important findings using this technology. And this is a high dimension where you can have more than 45, flow cytometry, we can push it up to 14, 16 markers, but here we have more than 45. But the challenge is, of course, then you're getting into math and statistics. You need to make sure that you can correct for multiple testing. So really powering your study is also very important. So this is where we used it, for example, first in immune-related diseases, in inflammatory bowel disease. This is from my PhD student that looked at mass cytometry using for IBD, as well as with the European. He also published this immunity paper last year. With that, the question is, because we don't have access to the human in acute pancreatitis, what can we do? So we go back to our animal models, and here we look at both local and distant. The reason we do local and distant is so that we could use perhaps these blood biomarkers or blood phenotyping that could help us. And, in terms of choosing the panels, even though you have 65 markers or 100 markers that you can use, there are more immune cell types than there is markers that you can choose. So this is where hypothesis-driven and how you use these technologies is very important. This is from Murley's study. And again, he has a poster distinction and we'll be presenting it on Friday. So please go to his poster where he has been able, using these types of models, these are experimental models. The top one is the acute pancreatitis in Cerulin, was able to show that were novel monocyte and macrophage subsets, and doing so also in severe pancreatitis where there isn't healing, where you have high mortality, he was able to differentiate and identify novel subsets. And this was just recently published in Gastroenterology. The next one is how we are using single cell RNA sequencing. And as I mentioned, working with Melina Bellin, where we have access to these pancreas samples. And it is very important also to have a control in anything that we do. So you need to have the same control. It's not good enough to have a surgical biopsy or a surgical resection of a chronic pancreatitis or other types. So we went to organ donors who also donate their islets, goes through the same procedure, because that's important. And this is why we worked with UCSF. So we get the controls from the UCSF and the chronic pancreatitis from Dr. Bellin. Here's the single cell RNA-seq. I think it has been described already in this conference. And site-seq, we also added site-seq. This is where you could do antibody linkage so that your single cell is not only on transcriptomics, but also you have an ability to really differentiate those immune cells. So we combined site-seq with single cell sequencing, and we also did a TCR at the single cell level. Remember, I mentioned in her previous paper, Bomi was able to find differentiating immune responses, but we didn't know them in details. And so I'll leave at that to her poster. Sorry, how did I get to this point? Apologize, kind of skipped a few of the slides. Can I go backwards, please? Yes, so this is one of her analysis, and please attend also her poster, which is today. The remarkable thing here is that if you look to the one that is on the right-hand side, you see this cell clustering that in fact idiopathic and hereditary pancreatitis also in the UMAP are quite segregated, and the clustering is quite different, suggesting that there is a difference at the immune cell level, even at the single cell level. And what's interesting also is when you look at the T cells, there is also a difference, not only in the number of T cells, but also the phenotype of those T cells, as well as the signatures, because this is transcriptomics, you could also go into more in-depth analysis. So here I summarize some of her findings, but please do go to her poster and she'll be able to tell you that she's identified an axis that seems to be very important in hereditary chronic pancreatitis, which is the CCR6 CCL20, which is the ligand, which is expressed on monocytes, and the CCR6 is expressed on T cells. Why is this important? Because we can block it. There are CCR6 antagonists, so we can study it. So potentially this is an important signature that she has uncovered. On the table is where we see, in fact, there is specific TCR sequences that seems to segregate hereditary pancreatitis. So there is a specific immune response at the T cell level. Maybe there is a specific epitope, even though we don't know what that epitope or antigen is. This is very important insight into that. So going on to why is important to do histology, Spatial cell analysis is important. As you know, the single cell, we sort them, we do many different things, but we don't know where they are located architecturally. And it's also important to do in situ so that you have not manipulated the antigen. But as you know, doing immunofluorescence has its challenge because there is limitation in the number of things that you can use. So there is this codex, which is also called a multiplex imaging, similar to CyTOF. But what's nice of this is also pioneered at Stanford in Nolan Lab. You can use a microscope that has actually three, just a regular type of microscope, and there are now antibodies. The reason you could do 62 antigen basically is because it's based on these oligo binding type of these antibody that are linked, unlike CyTOF, which you have heavy metals. So really important technologies, and what this tells you is also a neighborhood analysis. So you can even do single cell type of analysis at a signaling. So here is also from Bomi with other collaborators, other labs at Stanford, looking at a minimum spanning tree where you can see where these immune cells are located, but also what they are doing or who they are communicating in the neighborhood. So you could do this similar to CyTOF type of analysis that is shown on the right-hand side, which we call MST or minimum spanning tree. And she can explain that. I just want to touch upon briefly also on this that wasn't mentioned as Clarity is the another tool for those especially who study pain, for example, because you can get beautiful images and beautiful analysis. This comes from Deserov's lab at Stanford, which we've tried. And here's one of my instructor in the lab who is looking at intestine tissue, looking at these neuronal pathways because it's unblocked, so it's an intact whole tissue, and you could do this Clarity, and you can get very nice granularity. And this is my last slide, I believe. Just summarizing then what I just said is that we have great in-depth tool in immunology to decipher disease heterogeneity, identify novel functional subsets that can be used for monitoring, diagnosis, or therapy in the future, but it needs to be driven by the biology. It needs to be, there has to be an understanding of the technical and non-technical limitations and when to use it at what context is very important. Statistics is important because you need to power your studies. Otherwise, you do this, you will not find any significant difference because you have to correct for the multiple corrections or FDR. The next steps, of course, that remains from all these studies that I've shown you is we need to validate and determine the functional significance. And this is from high also, we need to take it to a thing that every lab can use. So once we validate and identify the most important signals, then one can transfer to CLIA type of flow type of low cost that can be used. Again, I want to acknowledge the lab. I just highlighted in burgundy here the people that I just mentioned, the work. And this is because my lab worked in other areas as well. This is very pancreatic centric. And there's also my collaborators on the right and the funding as well as the course that we used. And I will leave this for advertisement here for my, and to acknowledge also the people who did the work. Thank you.
Thanks very much. Questions will be at the end. Our next presenter is a virtual presentation. Anil Jaga from Cincinnati Children's, who's a professor in pediatrics, will talk about using secondary analysis of existing data sets.
Morning, everyone. First of all, I would like to thank the American Pancreatic Association and its organizers for inviting me to give this talk. The title of my talk is Secondary Analysis of Existing Data Sets to Decipher Drug-Induced Pancreatitis. So before I begin my talk, I want to make some disclaimers. The first one is this is not a hypothesis-driven study. What I'm going to present is primarily to showcase data-centric and data-driven workflows. I'm not a clinician, I'm a bioinformatician. And importantly, do not use any results presented here to make clinical decisions. And given the audience, the nature of this conference, there are no equations, and I try to minimize or don't use any informatics lingo in slides or the talk. And lastly, what I'm presenting is work in progress and everything is unpublished. The two primary goals for this presentation. First is to demonstrate the power and utility of secondary analysis, taking the existing data and then how we can repurpose, recycle it, reuse it for knowledge extraction and testable hypothesis generation. And importantly, as I mentioned, I'm not a clinician, I'm not a pancreatitis person, but I really look forward to the feedback and collaborations that can enable the validation of our computational discoveries and eventually translational research. I need to make some disclosures. So I'll begin with a introduction of the data sources, specifically the ones which we use for this presentation. And then I'll present two use cases. The first one would be how we can use existing data, mainly transcriptomic data, to characterize the drug-induced pancreatitis. And the second use case would be how we can use the animal data from the animal models, or again, the transcriptomic data for potential drug discovery, mainly drugs or compounds which could aggravate or mitigate pancreatitis. And this we are going to use both the omics data and also real-world data from FDA and electronic health records. So starting with the two broad categories of the resources we have used. One is the omics and the other one is the clinical data. So from the for the omics category, we use the expression data from NCBI's GEO, Gene Expression Omnibus. And for the annotations, the gene annotations, the single cell transcriptomic signatures, we use the top gene developed at Cincinnati Children's. And we also use the molecular signature database from Broad Institute. And for the drug discovery, we rely heavily on the connectivity map database, NIH-funded resource, which has about covers about 20,000 molecules and have signatures from different cell lines. And for the clinical data, we are using the FDA's data, which is the FAERS adverse event reporting system. And that has a collection of all the side effects related to the prescription medications. And the AIRSMINE is the application we have developed to mine essentially the FDA's side effects data. And AIRSMINE currently has about more than 14 million records. And the data range, what we have used is from the 2004 to 2020. And the last one is the Trinetics, which is a global health research network. What it has is close to about more than 80 million electronic health records from covering about 55 healthcare organizations throughout the United States. So except for Trinetics, all the resources I mentioned are open source and they can be accessed online. You don't need to install any application on they're web-based. So we'll start with the first use case where how we can leverage the existing omics data and then potentially formulate some mechanistic hypothesis. So we started with 4 well-known drugs which are known to implicate or known to cause pancreatitis: Azathioprium, didanosine, valproic acid, and asparaginase. So we went through the connectivity map database and then pulled out the signature. By signature, what I mean is the genes going up and down when treated with any of these compounds. And these signatures were based on different cancer cell lines. So for this study, I used A549 cells, except for the asparaginase, which is not in the connectivity map database. We used a publicly available published data set, which is based on the leukemic cells. So we took those top genes, like top 1% differentially expressed genes, genes going up and going down in each of the compound, and then ran it through the molecular signature database. See, the question we wanted to ask was, do any of the known drug-induced pancreatitis drugs have an off-target effect? And if on the pancreatic cells, and if so, what cells are affected? And the data set we use for that is from the human pancreas single cell data. So once you ran it through the molecular signature database, and then what you see here is the top 10 enriched gene sets. And each row is the single cell data set. And then what you see the column here are the genes from these compounds. So for example, Azathioprium downregulated from the 125 genes, 26 genes are expressed or enriched in the ductal cells, and 14 genes are enriched in the SNR cells. Likewise, the diadenosine, valproic acid, asparagines. So what was striking was among the so we were screening against the 671 data sets, which were covering heart, GI, pancreas, liver, and we could see that among the top 10, they do affect the pancreatic cells, and specifically the ductal cell seems to be a common theme across all these 4 drugs. And then among the genes, we could see many of the genes which are already have literature evidence or support associated with the pancreatitis or pancreatic injury or chronic pancreatitis and so on. Then this was with the genes which are going down when treated with any of these 4 drugs. And then the opposite was like what are the enriched cell types when in the genes which are upregulated when treated with any of these 4 compounds. And again, we could see the same trend where among the top 10 were the pancreatic cells, especially like if you see, for example, Azathioprium, the very first one, 21 genes of 124, 130 are from the ductal cell population. And asparagines was a bit surprising where we could see that among the 10, multiple cell types were from the pancreas, and there was an endothelial component, the delta cells and the beta cells and so on. And likewise the same gene. So ATF3 was in there. I think it was in the asparaginase signature, but we could see many of the known candidates previously implicated in the pancreatitis. So next we wanted to ask was if each of them are hitting on the pancreatic ductal cells, do they have any overlap? Is there any commonality between them? So surprisingly, the answer is no. So even though they are hitting on the ductal genes enriched in the ductal cells, they seem to be very specific to the compound. So as you can see in the 4-way Venn, genes downregulated following the treatment of any of the 4 compounds and the upregulated, we hardly see any overlap between them. And then we also wanted to see whether the genes which are down, for example, in the valproic acid, are they upregulated in asparaginase or vice versa? And again, the answer is no. They seem to be very specifically down or up, but again, very specific to the compound. So there was a recent study published in the eLife where they have reported identifying close to 6 or 7 subcategories within the ductal cell population. So that's something we want to, we're going to see whether these, they are actually hitting upon a subcategory of the ductal cell population. And what was another interesting thing which I have not shown here is the CFTR gene seems to be interacting with multiple members of this, each of these drugs. So next, that was the single cell, and then we used our database top gene to see what's the biological relevance, what pathways are enriched, what are the biological processes downregulated by these drugs. So what you're seeing here is a network view, mainly using the genes downregulated by any of the 4 drugs. And then the blue ones are the biological process enriched. So you could see, for example, the tissue remodeling, downregulated, a response to wound healing, and response to epidermal growth factor, and there are a lot of immune genes and so on. Then among the upregulated genes, what you see here, the orange are the upregulated genes following the treatment with any of these 4 drugs. We could see an ECM component in the diadenosine and some in the Azathioprium, and then in the asparaginase, we saw the retinoic acid receptor signaling, angiogenesis, and endothelial, which was reflected from the single cell data. For the valproic acid, we didn't find any enrichment in the upregulation. So by combining this, when I mentioned about the mechanistic hypothesis, is when we start digging deeper into it, then the single cell data and the pathways and potentially the protein-protein interactions centered on the CFTR or other known pancreatitis panel genes, they can start, we can formulate the hypothesis and which can potentially testable. So in the second part, I want to cover is how we can use the existing data for discovering potentially small molecule modulators which can either aggravate or mitigate pancreatitis. So here we use a concept connectivity mapping from introduced by Lam et al. In about 15 years ago. So by nature, what the connectivity map does is it looks for a reciprocal connectivity. So it can start from a biological state where it could be a mouse model or it could be a cell line or a human patient data, and start with the transcription data set, microarray, RNA-seq, single cell, anything, genes going up and down. And then you compare that against a drug database or a small molecule library, where you have the differentially expression signatures from each of the compound. Then you look for a pattern matching or a correlation. So genes going up in the disease, if they're going down, then that's a strong negative. Whereas genes going up and down, they're also going up and down the same direction, that's a strong positive. So we can use that in two ways. One is the compounds that are transcriptionally, like, for example, azathioprium or didanosine. Take those drugs, signatures, and then run it through this library, and then look for compounds or new investigational molecules, phase 2, phase 3, early stage compounds, and then see which one could be likely or highly similar. Could they be new candidate drugs for drug-induced pancreatitis? The second one, more interesting one, would be to see for compounds which are opposite to the signatures, for example, azathioprium, and then what could be anti-correlated to that, or just use the mouse models or human patient data or any other data representing pancreatitis and look for candidate therapeutics, mainly the genes which are negatively correlated with the disease state. So we started for this study, we used the Cerulean model. We used four data sets. And what you see here is a Venn diagram representing the four data sets. So on the left side are the genes which are going up, and on the right side, you have the genes which are going down. And there are 127 genes which are consistently upregulated in all the four studies. Then on the opposite side, you have 16 genes which are conserved or consistently downregulated in all the four studies. So we took that signature because that seemed like a robust signature represented by 4 independent studies. And we used that 127 up and 16 down, and then did what I showed just earlier, connectivity mapping, submit to the database and get the compounds, list of compounds which are positively and negatively correlated strongly to the Cerulean model signature. And here is a heat map view of that list of compounds. The blue means they are negatively correlated, and what I'm showing here is the significant top 40 or up and 40 down. And among the negatively correlated molecules where what attracted our attention was some of the approved drugs, well-known drugs in the market, well-studied and so on. 2 of them were the calcium in the top. Actually, Varapamil was top ranked. And then when these are based grouped on the categorized based on the class. And there were 2 drugs which were calcium channel blockers. And then we also wanted to see, like for example, if can we use any of the driver genes or transcription factors, and then look for the small molecules which could be either positively or negatively correlated. So for here, what we did was we took the ADF3 knockdown signature. Again, by signature, I mean the genes which are going up and down following the knockdown of ADF3. This again from the same database, using the A549 cell lines. And then the question was, what are the compounds which are transcriptionally similar to ADF3 knockdown? And then again, we have the positively correlated and negatively correlated compounds. And among the positively correlated compounds, which are similar to what you get from the ADF3 knockdown, among the top 10 was the Varapamil again, and there was another calcium channel blocker. And then I'm going to show subsequently where when we mined the electronic health record data, we could see a potential benefit for the Varapamil. And another compound which was interesting was the bromhexine, which is a known mucolytic. And since the ductal mucus obstruction is reported to be one of the early defects in chronic pancreatitis, the hypothesis, the therapeutic hypothesis would be, can mucolytics like bromhexine or other mucolytics could be of therapeutic benefit. So this was purely based on the animal models or the cell line data. Now we wanted to see whether we can replicate these findings in the real world data. So the question was whether Varapamil, so we took the example of Varapamil, and then whether it has a therapeutic potential for pancreatitis or drug-induced pancreatitis. So we first started with the FDA's adverse event data. So we used the AIRSMAN application. The question was mining the records or getting all these about 14 million records, get all the patients who are on Azathioprium but not taking Varapamil, and then compare them with the patients who are on Azathioprium plus Varapamil, and then look at their side effects profile. Mainly what I'm showing here is the pancreatitis related side effects, abdominal pain, amylase increased, lipase increased. So what you can see is patients who are not on Varapamil, who are only on Azathioprium, seems to have most of those side effects. But the patients who are taking Azathioprium and also Varapamil, they seem to have a lower risk for most of these side effects. So at least based on these safety signals, it appears that patients who are have a concomitant use of Varapamil have a reduced risk for pancreatitis or pancreatitis related traits. Now, since the FAIRS data has its own issues with the noise or over-reporting confounders, we wanted to see whether we can replicate that with the electronic health record data. So we use the Trinitex database, which I mentioned earlier. And then here the question was, does Varapamil mitigate the risk for Azathioprium-induced pancreatitis? So the query is again the same way. Patients who are on Azathioprium alone, they are not taking Varapamil, and then patients who are taking both. And then looking for the outcomes is the acute pancreatitis and chronic pancreatitis. We use the ICD codes which are in the database. And then we could see that again, the trend is the same, where there seems to be a reduced risk in the patients who are taking Varapamil simultaneously along with Azathioprium. And then to the application has supports the propensity score matching, where you can do the cohort matching for age, race, sex, and potential other confounders. The other question we want to ask is, if Varapamil is beneficial, are there any long-term benefits of using it in the acute or the chronic pancreatitis patients? So we repeated the query, but this time we only focused on patients who are ICD coded with acute pancreatitis or chronic pancreatitis. And then the outcome was what is the risk for them to be diagnosed with either pancreatic cancer or more specifically the pancreatic ductal cancer. So we again use the ICD codes. So it turns out, and this was again done after the cohort matching. So as you can see, before matching and then after age, sex, and other traits, the race matching, they were, you cannot see them, but they overlap together. And then again, we could see that there was a decreased risk for pancreatic cancer if the patients are on Varapamil, along with if they are already diagnosed with a chronic pancreatitis. Then the question, the next thing we wanted to ask is if there's a potential benefit we saw in both with the Azathioprium and also in the pancreatic cancer. Is that limited to Azathioprium or does it extend to other pancreatitis drugs? And we repeated the query this time again with the FAIRS data.
Just because we're running so far over, I was gonna have the folks stop this talk, and we'll be an opportunity to ask questions at the end as well, just because we're gonna run out of time for the third speaker. I apologize.
Okay, so moving on to the speaker we have next, Dr. Sohail Hussain from Stanford. He is endowed professor and the chief of the Division of Pediatric Gastroenterology, Hepatology, and Nutrition. Sohail will be speaking to us today on using open source data to complement the study of pancreatitis.
Great. Thank you, Maisam and Chris. I'm going to try to make up for some of our times to allow for additional questions. And at the outset, I wanted to acknowledge assistance for this presentation from lab team member Toshi Sato. What's the purpose of this last talk on emerging technologies? You heard from Aida brilliantly about generating large data sets using multidimensional tools, and this generates the open source material. You heard from Anil in this last talk about existing data sets to decipher drug-induced pancreatitis, and this was a practical use of open source data. Well, my talk will be a wet lab and clinician researchers' perspective about how to get immersed in open source data. Call it data mining for dummies. I would invoke the cliche, Houston, we have a problem. And not only are we over time, the other problem is that the traditional pathway of medical discovery is that you first generate a hypothesis, and then you go about testing it with experiments in the lab or in human populations, right? Well, the problem is that this is time-consuming and costly. Meanwhile, there's an explosion, as shown in this cartoon, of open source data relating to virtually every field in science and business for that matter, including in medicine. Most of it is freely accessible on the internet. In fact, federal agencies and many journals require it. There is a second problem, which is of information overload. Accessing the vast amounts of data onto a usable format is daunting. It's difficult to know how to get started. This, however, is a major opportunity to harness open source data to cost effectively accelerate medical discovery. Big data is a fast-growing global commodity with a bright forecast, and this comes right out of Forbes.com, a favorite journal. Well, here are some tips for working with large data sets, and this comes from a recent review. Automate rather than manually comb through your data, including for data quality assurance, validation, data repair. Document what you do, your workflow, operating environment, the versions you use. Plan out your computing time and record metadata. Avoid downloading the raw data if possible, and use online platforms where available. Get the training. About a decade ago, a group of data users came together and over a few years put together what's called the FAIR data principles. And it was meant to enhance the ability of machines to automatically find and use data in addition to supporting its reuse. And they came up with the acronym FAIR, which refers to easily finding data, easily accessing it, making it interoperable across systems and workflows, and of course, reusing it for the benefit of all. And based on these principles, there is Omics Discovery Index, or Omics DI, which is an open source platform that can be used to access, discover, and disseminate omics data. And what's shown on the left is the workflow for this particular website, which is that they take public repository data and then they put it into a usable format, in this case, an XML file. And then that XML file is validated, annotated, indexed, and then available for a program interface or API. And often these are online browsers. Currently, Omics DI, for example, has 2.3 million data sets available to anyone, and it's growing. So let's get immersed in open source data use, break the ice, if you will. I'll walk you through step by step through a real case example using open source data to decipher the problem of pancreatitis due to the drug asparaginase. And it'll be different from the example that Anil had showed, although he was a major collaborator and a go-to person for this work. Drugs are a leading risk factor for acute pancreatitis. If you define, that is, if you define drug-induced pancreatitis as a non-exclusive risk factor, not just a diagnosis of default. And here's some data in children from our own group on the left and that of MISM shown on the right. Asparaginase is among the most common drug identified to induce pancreatitis. And here are a couple of publications that indicate that. Well, asparaginase is a crucial chemotherapeutic for acute lymphoblastic leukemia. However, 5 to 10% of users develop pancreatitis, and 1/3 develop severe disease. In a recent serum metabolomics screen that Olivia Tsai will talk about in a presentation in the next session, we had identified in a case control group that a single exposure of asparaginase among pancreatitis cases led to a reduction, surprisingly, in the micronutrient precursor of vitamin A, the carotenoids. And that's shown in the volcano plot of fold change after a single exposure to asparaginase in the pancreatitis cases. So these were intriguing data, and we wanted to ask whether we could complement them with open source data. And so we turned to Omics DI, searched acute pancreatitis. It yielded over 1,000 results. For various reasons, we focused on transcriptomics data. And because of the low numbers, we decided to either manually glance over the descriptions, or you could refine the filters just like in any other filtered search. We identified one transcriptomics data set, which in this case came from Matthias Lohr at the Karolinska from 2018 in a publication in Pancreatology. And fortunately, it's also linked to an analytical tool called GO23. And so when you press on the tool, you get what's shown on the right. You see the actual sample data. And you now need to just specify the groups that you want to analyze. In this case, we specified controls and pancreatitis. And it automatically generates for you. And this is the beauty that I really want to express to you. And if that's the main message that you take out of this, it's that you automatically generate data within seconds, if not minutes. And here are 9 automated visualization plots that come to you, along with a list of the top 250 differentially expressed genes. And so let's go through some of those plots. First, the box plot. The box plot tells you that the data are normalized and cross-comparable. That's check number 1. Then there's a volcano plot, which is generated that demonstrates that there's impactful statistically significant differences in the gene expression between the two groups. And this is an interactive plot where you toggle over each of the dots and are able to further drill down on the genes. Check number 2. Check number 3 is that you get a UMAP, which is like a PCA. It shows that the samples are related or unrelated to one another. Check number 3. And then beyond those global analyses, you can drill down on the individual genes of interest. In this case, we were interested in vitamin A related genes, such as all trans-retinoic acid induced differentiation factor. And you can see that it's reduced in cerulean pancreatitis, as shown on the right. Well, to fast forward, we eventually identified 2 transcriptomic data sets using this process. One that I showed you for pancreatitis and described on the left, and another for asparaginase exposure in leukemic cells, shown on the right. And then using those 2 data sets, we performed, I should say, Anil performed an in silico chemical perturbin screen. And we found that the retinoid analogs are among the top hits predicted to prevent or reduce pancreatic gene upregulation. And this was just an example to show the power of open source data to complement our work. The main points from this example are that we're able to use the FAIR principles. We're able to easily find our data sets of interest using omics DI. We were able to access them and analyze them using GO2R, and they were interoperable among 2 data sets, for example, on a perturbin analysis. And this really highlights the reusability of an open source data set online without having to download the data. We analyzed the raw data as if it were our own. And we did this not within weeks or months, but within minutes or hours and without cost other than your own cost, which is why funding agencies still need to fund this work, of course. Here are the acknowledgements for the asparaginase work. And I'd like to highlight, in addition to Toshi Sato, Olivia Sai, who's here and will be presenting in the next session, and our collaborators from Cincinnati, including Mayur, Maisam, and Anil. In summary of using open source data to complement the study of pancreatitis, I think I have 1 more minute remaining. Thank you. Open source data analysis strengthened our lab findings. It provided the impetus to pursue a role for vitamin A supplementation to prevent or treat asparaginase-associated pancreatitis, and perhaps the first precedent for acute pancreatitis. And so get immersed in open source data. Get the training, take courses locally, online, or on-site. Live vicariously. Send your trainees or lab people. Learn coding. And now it's much easier to learn coding than it was before. Partner with data scientists and frequently ask whether your scientific inquiry can benefit from open source data. Here's some open suggestions as we brainstorm for the APA, perhaps training platforms, crash courses, or sabbatical applications on open source data use, getting more data scientists as members of the APA. These are just personal opinions. And using open source data to complement your work and to enhance, therefore, the exocrine pancreas field. And so with that, thank you for your attention.
Thank you, Sohail. If we can invite Aida also to the stage and get Anil on the virtual platform. We apologize to Anil for having to cut the time. Indeed, it was a very energizing session, at least because it applies directly to a lot of the summaries of talks that we had. And if I may reflect for a second, the biomedical informatics field and data scientists need us as clinician scientists and basic scientists who work in the knowledge content of pancreatology, but we do need them. This is the day and time of really huge data sets and data management through machine learning algorithms. And in fact, it is the future in a lot of people's mind and the new generations. So we do have some questions, and I see Dr. Miklos Sahintat. Please go ahead.
I just want to thank the presenters for this really
In a lot of people's mind and the new generations.
It's not me, I don't think.
Is it on?
In a lot of people's mind and the new generations.
Is that you, Chris?
I'm trying here, team.
Is it on?
In a lot of people's mind and the new generations.
Is that you,
I
Chris?
can tell people are tired from my voice, but
I'm trying here, team.
Anyway, so this was a fantastic session, and I learned a lot. It was really great. I think so here we have to thank you because you were driving this session.
I can tell people are tired from my voice.
Getting
But
the best speakers. But anyway, so this was a fantastic. I'm an experimental scientist. It was really great. And really, the value of these activities to generate driving.
I can tell people are tired from my voice. But
And I didn't see any examples.
But anyway, so this was a fantastic.
I'm an experimental
scientist.
scientist. So I think that was a very interesting session. Creation of theories we can, follow up. Maybe if you want to comment on that. Yeah, thank you, Miklos. I mean, in the interest of time, obviously you use these data and then of course validate them with actual experiments. In fact, there's an interesting article as I was preparing for this talk from a few years back, arguing the counterpoint, which I think data, and then of course about 10 years ago, which was that data scientists were called data parasites, which is that they would just use other people's data to, generate their own. But I think based on many papers in very high impact journals that complement the actual experimentation, this is no longer considered a term.
Superb presentations, very enlightening, and I have no words but to thank you to bring these new technologies up to the table for all of us to understand and comprehend. My questions about this, and I have a few of them, relate to first cause, effect, and bystander. So whenever we see something going up or coming down, that may be just a reaction to the insult, or it may be the cause of the insult, or it just may be a bystander which has nothing to do with the cause. The second part is most of this is, if you look at disease processes, there's a temporal association, that something started, but then something happened later. So there is a temporal association that takes place, which may be relevant to deciphering whether it is actually causal or just an effect or bystander. So how do you make these distinctions in large data sets?
I think I'll start and then maybe Sohail, you can follow. Vijay, you're right. I think this is what I meant also by context. The context matters in terms of how I think the big data set, what we have to make sure also, it's if you put garbage, you get garbage out, right? So the quality of data matters and how it was, so one needs to be very careful how it was collected, is it, so the context really matters and you need to validate it, obviously. So one has to use a separate discovery and separate validation. So.
if I may ask Anil also, thank you, Aida. This is exciting, and I totally agree with you. Anil, through our collaborative work, if you can comment, this is a great question from the audience on how would you establish causation from big data sets? And when we did the exercise and used the Bradford Hill criteria and the 9 factors that we use, we actually make a good case how big data sets could confirm or refute what you find in the lab and in clinical research. If you just want to comment on that.
Thanks, my son. And I take this time to apologize. I think I've sent a long version, so I'm really sorry for that. Sincere apologies. So I think that's a great question. What Aida said is very relevant about the garbage in and garbage out. And then what we found very useful was the collaboration, like for the informaticians collaborating closely with the wet lab and also with the clinicians. that helps a lot, especially for the other question about the confounders. Often what we see a strong signal could be just a confounder, or there could be another factor, but that helps only if we have a domain expertise, and that can help in addressing some of those issues.
My second question is specifically for Anil. You mentioned adenosine in the drugs that you tested. This is very clinically relevant because if you look at HIV, for which adenosine is a NRTR, or nucleoside reverse transcriptase inhibitor, till 1996, when adenosine was largely used for HIV treatment, the risk of pancreatitis in HIV was very high. And adenosine has cousins like stavudine or azathioprium, which was the first drug for HIV. So pre-1996, books like Harrison's used to publish the risk of pancreatitis is in over the common population. I remember that. But then comes heart in 1996, and pancreatitis has dramatically come down. Now, that does bring up a question that there are other drugs too that cause pancreatitis apart from adenosine. Second, are the new drugs protective, like the protease inhibitors that came on after heart? So have you looked at that aspect, or would you like to look at that aspect in your large data sets? Because those are all available.
Yeah, so that current PhD students' work is looking at the polypharmacy aspects and also the temporal nature. So that's where the Trinitex helps, where we can backtrack and look for last 10 years, last 5 years, and then what the secondary drug is was used. So where the genomics and then the real world data would help drug-drug interactions, but it becomes complex as the most of the current approaches, they focus on only 2 drugs at a time, but it could be multiple drugs, 4 or 5 drugs, and then over the counter. So yes, it becomes a real big data and a machine learning problem. But the deep learning based approaches, what we are currently working on, that helps a lot.
Hi, my question is for Anil too. So Anil, I see that you use Varapamil, and Varapamil is a blocker, it's a channel, calcium channel blocker that affect both heart as well as blood vessels. I just wonder if you wanted to study in the future just more specific calcium channels blocker that are specific for blood vessels, like for example, nifedipine.
Absolutely, I think that's where I think one of the things where we are looking for more feedback and more suggestions, and so we can. The only limitation is the data. Most of the data is available is from the cancer cell lines, but definitely, if we have a list of all the calcium channel blockers, we can do that.
I just had a sort of a philosophical question for Aida and Sahil to as I'm thinking about this. There's an approach which is sort of the data first and see where it leads, and there's an approach which is the clinical question first and then try to ferret out the answer. And you guys have, I think, approached various questions in both ways, and I wonder if you could just give it your perspective on which strategy is. I mean, this is a meeting that brings together basic scientists and clinicians. It's one of the few that does so. And so from the clinical side, it's sort of, how should I be thinking about this, and how can I interact with you in a way that's, leads to something that's clinically actionable?
Thank you, Chris, for that question. So I always like to think the question rather than the tool, and that's, but definitely I think it's where we meet halfway. And I think there is a value for example, we do unbiased, especially I think a lot of some of the discoveries, especially when you don't know the disease and there isn't enough data, you do unbiased analysis. It takes you to a path, but you still need the biology in there. So I think I think it needs to be collaborative and I think it's an approach that I think biology needs to be there, but we also need the data scientists to help us decipher, because it's really huge amount of data, the mathematics, and things are in that field is also developing, right? Just as much as we think our biology is heading. So I think there needs to be a team size. I think this is something we have to think about rather than individual. Of course, there is importance about individual thinking and independence, but we also need to bring the two areas, because I think at the end, we want to make sure there is significance to the question or to the biology, and how do we interpret the data. So we need each other. The way I see it is we need to work together.
Can I just add to that? Thank you, Aida. For example, with the vitamin A story that hopefully you'll hear more about, we had known about these data from enrichment studies for about 2 years, and I really didn't know what to make of it. So it was really hypothesis generating in that case. And then I met through MySum Anil, and one day he said, oh, well, we found vitamin A in our FDA adverse event health records as being anti-correlated to pancreatitis. And then a light bulb went off that maybe there was something here. And then I found a nutrition faculty researcher as part of a consortium who had dietary intake information. And so I guess you need to do both. You need to sort of keep an open mind and look at your unbiased results. And if you've got enough sample size, that's also the question. If you've got low sample size, be careful about interpretations. And that's particularly true for genomics data, but for any omics. But if you've got the sample size, then let the data guide you.
Another thing I would just add is the heterogeneity in the human, right? So when, especially sometimes when we do, of course, hypothesis-driven questions are important, and we are focused, and when we do, because we do both animal and translational human work, is that when you try to translate it, you go into a very focused single species you're trying to get to. So there is an investigator at Stanford also who says, as human beings, we are diverse species. So it's very important to, if you see something within different data sets and it's consistent, more likely that biology and signal is actually a very strong one. So heterogeneity and putting heterogeneous data sets together to try to understand is another. So there are many different ways. It just has to be handled by the right people who have the right training in that aspect.
And it seems we just have to somehow maintain an ability to talk to each other. As you listen to some of these lectures, as sort of the information, you're like drinking from a fire hose, and it's like, wow, how can I even think about that? They're using nomenclature I don't understand and can't remember, and it's impressive. Anyway, it was just terrific. And I love the story about Sohail, about you sort of like, well, then maybe this person, and then the nutritionist, and it's sort of all this team you would never think to pull together suddenly falls in place around these ideas. Just wonderful. Other questions from the audience or comments?
Well, I had one kind of a quick maybe question to Anil, and I don't know if you can answer it fully, but you being in an APA format, from your work on the molecular signatures, genes, cells, from the system biology approaches, what do you identify as the major knowledge gap with the data that's out there in terms of the libraries of transcriptomics in the field of pancreatology? What can scientists in this room know that you really, really need for us to enrich the data sets on so that we could do better system biology design studies in pancreatology?
I think that's a great question, and I'm not sure if I have the answer, but I think what would help is the reproducibility, because as you have seen in the presentation, this even with the cerulean model, we don't see much overlap. So that's it. And then the numbers, I think Sohail mentioned about the lack of numbers on the genomics. So high throughput, that would help. And the other one is the model. So I don't know if what would be the right model, even for the pancreatitis or the drugs. Would it be organoids or pancreas on chip, or should it be continued with the cerulean? So those are more, I don't know if that answers the questions, but I think that's where. And then I think to add to what Sohail said, we have lack of data on genomics, but there's a data deluge on the EHR or the side effects and so on. So I think somewhere hitting on the middle ground, that's what I think.
That's great. Thank you. Indeed, when you get data from data mining, it brings the question, what is the right preclinical model to verify it for verification? And is it human-based organoids or is it animal models in the lab? Do you guys have any comments on what would be some of the models that could be used for confirmation? I think you do both, right, in the lab experiments? Yeah.
I agree. Yes.
If no other questions, we'll finish. We have 5 minutes until the next, the Frank Brooks lecture starts, so please don't go far.